Hi Dr. Suganti and my fellow friends. My name is Nurul Aibati Ramlan, representative from Group 3. In this video, me and my team will introduce you what is yeast. So let's get started.
Yeast are eukaryotic and single-cell microorganisms classified as members of the fungus kingdom, which also include molds and mushrooms. The scientific name of yeast is Saccharomyces cerevisiae with a single-cell grow habit. Yeast can be contrasted with molds, which grow high fat. Yeast are also found on the surface of the skin, where they may live symbiotically or as parasites.
Functions of yeast. The products of these reactions have been used in first, baking, second, the productions of alcoholic beverages for thousands of years, third, as starter cultures in cheeses and bread, as well as wine, beer, and other alcoholic fermentations products. In addition, yeast can also initiate spoilage in foods such as yogurts, fruit juice, salads, mayonnaise, and is commonly found on plants, leaves, flowers, and foods as well as in soil.
How yeast work? Yeast is a type of good fungus that is involved in the fermentation process, the process of producing energy in cells anaerobically without oxygen. The main function of yeast is as a leavening agent or catalyst, causing the food to be hollow, soft, and fluffy. The yeast will take advantage of the sugar, carbohydrate content of the flour and release carbon dioxide, which will eventually cause airspace to form in the flour. The airspace that will cause the bread or dough to become lighter and fluffier.
Breeding process. Yeast reproduce by budding. During the process, a small bump, the bud produces from the parent cell. This enlarges and matures. The bud eventually breaks away from the mother cell to form a separate larger cell.
Biochemical pathway of yeast. Yeast have two pathways for ATP production from glucose, respiration, and fermentation. Both pathways start with glycolysis, which result in the production of two molecules of pyruvate and ATP per glucose. In fermentation, pyruvate is then turned into ethanol. In respiration, pyruvate is completely oxidized to carbon dioxide through the TCA cycle and oxidative phosphorylation. Group3 positive yeast at sufficient level of oxygen and glucose use fermentation and respiration simultaneously. The ethanol that accumulates in the environment can recycle for ATP production once glucose has been depleted.
Yeast fermentation. Alcoholic fermentation. The production of alcoholic beverage from fermentable carbon sources by yeast is the oldest and the most economically important of biotechnologies. Yeast play a vital role in the production of alcohol beverage because it can maintain beverage sensory quality. Anaerobic respiration in yeast is under alcoholic fermentation as a glucose broken down to produce ethanol, carbon dioxide, and some ATP. Bread fermentation. The fermentation of the dough made by the yeast in the most critical phase of making bread. Yeast not only produce CO2, other metabolites, and influence the final appearance of the dough, volume, and texture and the taste of the bread. Fuels and other chemicals. The fermentation process of substrates such as yeast were high interest of industrial level. These substrates allow the environment cost efficient production of the biofuels and other advanced chemicals to be reduced. Coffee fermentation. Yeast play an important role in the coffee production in the post-harvest phase. This performance can be done in two phases. Firstly, aerobically, in which the berries just collected are deposited in a tank and the yeast are allowed to act. The process is carried out under control of basic parameters such as time and temperature. Next phase is in anaerobically, which is absence of oxygen, where the berry is mixed with water in jar. The process to improve the organoleptic properties of coffee, refine its sweetness and control acidity.
The SEN1A is commonly grown in higher agriculture area in North Thailand at elevation. Chemical-based fertilizer are commonly used for the promotion of tea plant growth, which are also known to cause environmental pollution, soil acidification, heavy metal pollution, soil compaction, and changes in soil microbial population. The aim of our study was to isolate yeast strain obtained for reservoir soil of an acetia plantation in North Thailand to study their plant growth. For the reason, researchers have been interested in replacing chemical-based fertilizers with techniques that improve the application of beneficial microorganisms. They can support the sustainability of the agriculture industry and the environment. PGPN have been defined as microorganisms, fungi, bacteria, autonomously, and yeast. They can inhibit plant growth, improve plant nutrient intake, induce systematic resistance, and increases stress tolerance of one of the following mechanisms: nitrogen fixation, solubilization of insoluble mineral, and other. Promoting abilities funding for this research study can be asked to develop playground enhancing yeast as bioinocular to replace chemical fertilizers currently in use.
Okay, so how the experiment will be conducted? Step 1 is soil samples were collected from 3 different places of Assam tea plantation. The soil sample were kept in sterile plastic bag and carried to the laboratory on ice within 48 hours being collected and were processed for isolation yeast immediately. pH and electric conductivity of each soil is determined and recorded. Yeast strains were isolated. 1 gram of soil sample is placed in 99 ml of yeast malt extract (YM broth) that had been combined with 50 mg per liter of chloromethylnickel in 250 ml aluminium flask. Flasks were placed on orbital shaker with shaking at 150 revolutions per minute in the jug at 30 Celsius for 48 hours. After 48 hours of incubation, the dilution split plate of the method was employed with 7 serial dilution in 0.5 NaCl solution. After dilution, 0.1 ml of suspension was sprayed on YM agar. Combined with 50 mg per liter of chloromethanol, the plate were incubated at 30 Celsius and for 48 hours. Different yeast colonies were selected based on relevant morphological characteristics. The individual colonies were regrown at 30 Celsius on YM agar for purification. The pure strains were kept in 20% glycerol at 18 Celsius. So that's all.
Result 1. Yeast isolation and identification. The result review. The 11, 70, 40 strains were obtained. 2. A total of 40 strains were identified belong to 12 non-yeast species and 3 unrestricted species. 3. Under culture conditional or yeast strains were effectively able to grow.
Next, I will present related to yeast infection. The yeast infection that I will present focus on three parts, among which is a yeast infection in adults, the target of yeast infection that can occur in infants, and finally microorganisms found in yeast infection. Yeast can also cause infection in humans, regardless of adults and children. The yeast infection is usually more likely to occur in children, especially babies. This is because the thin surface of their skin can cause yeast to easily enter the bloodstream. One of the yeast infections that can occur in adults is itching in the vagina, while yeast infections that can occur in children are infections in the mouth, better known as oral thrush. Next, there are microorganisms that induce this yeast infection to occur. The microorganism is known as Candida albicans.
That's all from us from Group 3. Thank you for your attention. So bye.